What if I told you that right now, in laboratories around the world, artificial intelligence is dreaming up life forms that never existed? Not monsters, not mutants, something far stranger. Molecules. Tiny molecular machines built atom by atom that nature never thought of. Proteins that can eat plastic, proteins that can cure diseases we haven't even named yet, proteins that are quite literally impossible. Except they're not impossible anymore. Welcome to the story of how humanity learned to speak the language of life itself. The 50-year curse. Let's start with a riddle that tormented the smartest people on Earth for half a century. In 1958, two scientists named John Kendrew and Max Perutz did something extraordinary. They figured out the 3D structure of a protein called myoglobin. It was like seeing the blueprint of life for the very first time. They won the Nobel Prize, champagne flowed, everyone celebrated. And then reality set in. Because when they looked at this thing, they saw something that made no sense. The structure was, quote, almost totally lacking in the kind of regularities which one instinctively anticipates. That's scientist speak for, what the actual hell are we looking at? This protein was messy, chaotic. It looked like someone had thrown a ball of yarn into a blender and hit puree. And yet, it worked perfectly. This tangled mess could carry oxygen through your blood with the precision of a Swiss watch. Here's the problem. Proteins start as simple strings of building blocks called amino acids. Think of them like beads on a necklace. But then, within 1,000th of a second, they fold themselves into these insanely complex 3D shapes. And here's the kicker. The shape determines everything. The shape is the function. A misfolded protein isn't just broken, it's dangerous. Alzheimer's? Misfolded proteins. Parkinson's? Misfolded proteins. Mad cow? You guessed it. Misfolded proteins having a very bad day. In 1972, a scientist named Christian Anfinsen won his own Nobel Prize for proving something that sounds simple but was actually profound. The amino acid sequence of a protein contains all the information needed to determine its 3D structure. In other words, the code is there. We just need to crack it. And so began the 50-year curse. Levinthal's Paradox, the impossible problem. Let me introduce you to Cyrus Levinthal. In 1969, he did some math that should have made every protein scientist quit and become a florist. Levinthal calculated that a typical protein has approximately 10 to the power of 300 possible folding configurations. Let me put that in perspective. There are about 10 to the power of 80 atoms in the observable universe. That means if you gave every single atom in the entire universe its own universe, you'd still be short. If a protein tried every possible folding configuration one by one, it would take longer than the age of the known universe to find the right one. And yet, proteins fold in milliseconds, sometimes microseconds. This became known as Levinthal's Paradox, and for 50 years, it was like the scientific equivalent of that math problem in Good Will Hunting that everyone stares at, but nobody can solve. The best minds in biology tried everything. They used supercomputers, they developed complex mathematical models, they spent years, sometimes entire careers, trying to predict the structure of a single protein. And here's the really frustrating part. We knew it was possible. We watched proteins do it every single day in every living cell on Earth. We just couldn't figure out how. It was like watching a magician perform the same trick for 50 years, knowing there was no actual magic, and still not being able to figure out how they did it. Enter David Baker. And I have to tell you, this guy's origin story is almost too perfect to be real. In the early 1980s, Baker was a senior at Harvard, majoring in philosophy and social studies. Not biology, not computer science, philosophy.
This guy was literally trained to ask impossible questions. For a term paper, he wanted to write about protein folding. His professors told him not to. They said, and I quote, "No one really knows how it works." Most students would have picked another topic. Baker? He filed that as problem to solve later. Fast forward to 1993. Baker is now running his own lab at the University of Washington, and he makes a decision that seems, frankly, insane. He's going to solve protein folding using computer games. In 1998, Baker releases a program called Rosetta. It's designed to predict protein structures, but the calculations are so intensive that even supercomputers struggle. So in 2004, Baker does something brilliant. He releases Rosetta at Home, a program that lets anyone donate their computer's idle processing power to help solve protein structures. By 2008, nearly 200,000 volunteers had signed up. Regular people just letting their computers crunch protein folding problems while they slept. But Baker wasn't done. Some volunteers said they wanted to actually see what their computers were doing. They wanted to help guide the folding themselves. So in 2008, Baker released Foldit, a multiplayer online game where ordinary people compete to fold proteins into their lowest energy states. And here's the wild part. Foldit players, people with no formal training in biochemistry, started solving problems that had stumped scientists for years. In 2011, Foldit players helped decipher the crystal structure of a retroviral protease that causes AIDS in rhesus monkeys. This structure had stumped scientists for 15 years. Gamers solved it in 3 weeks. Let that sink in. A problem that PhDs couldn't crack for 15 years was solved by people playing a video game in their underwear. But as revolutionary as Rosetta and Foldit were, they were still just better ways of doing the same thing. They were essentially still brute forcing the problem, trying millions of configurations until something worked. What happened next would change everything. The AlphaFold earthquake. In December 2018, a small team at a London-based AI company called DeepMind entered a competition that most biologists had never heard of. The competition was called CASP, the Critical Assessment of Protein Structure Prediction. It had been running since 1994, basically the Olympics of protein folding. Every 2 years, teams from around the world would submit their best computational predictions for proteins that had been experimentally determined but not yet published. The organizers would compare the predictions to the real structures and score them. For decades, progress had been glacial. Each competition, teams might improve by a few percentage points. Incremental gains, slow, painful progress. And then, DeepMind showed up with something called AlphaFold. AlphaFold didn't just win CASP 13 in 2018, it dominated. It achieved a summed z-score of 52.8. The second-place team, 36.6. It wasn't even close. But here's the thing. The DeepMind team wasn't satisfied. They knew they could do better, much better. Demis Hassabis and John Jumper went back to the drawing board. They spent 2 years completely rebuilding their approach. They added geometric constraints because atoms have specific sizes and bonds have specific angles. They developed new ways for the network to learn from limited data. They created mechanisms that paid special attention to amino acids that ended up close together in 3D space, even if they were far apart in the sequence. And in November 2020, at CASP 14, they unleashed AlphaFold 2. The results were, there's really no other word for it, absurd. AlphaFold 2 achieved a median GDT score of 92.4 out of 100. For context, 90 is considered roughly equivalent to experimental accuracy. AlphaFold 2 wasn't just predicting protein structures, it was predicting them as accurately as X-ray crystallography, the gold standard that took months or years and cost hundreds of thousands of dollars. For the most difficult proteins, those with no similar known structures, AlphaFold 2 still scored 87. The next best method? It wasn't even in the same universe. John Molt, the organizer of CASP, announced at the end of the conference that AlphaFold 2 had largely solved the protein folding problem. A 50-year grand challenge. Solved by an AI. But here's what makes this story truly remarkable. DeepMind didn't just keep this breakthrough to themselves. In July 2021, they published their methods in Nature and released the source code. Then, in partnership with EMBL-EBI, they released the AlphaFold protein database with predictions for nearly every known protein in the human genome. And in 2022, they expanded it to over 200 million proteins, covering virtually every
organisms whose genome had been sequenced. Let me put this in perspective. Determining a single protein structure experimentally used to take a PhD student their entire graduate career. AlphaFold 2 could do it in minutes. And now, anyone with an internet connection could access predictions for 200 million proteins. What would have taken hundreds of millions of years of experimental work was now available at the click of a button. Over 3 million researchers in more than 190 countries have used AlphaFold. More than 30% of AlphaFold-related research focuses on understanding disease. Scientists have used it to study antibiotic resistance, to design enzymes that break down plastic, to understand cancer mechanisms, to develop new vaccines. The 2024 Nobel Prize in Chemistry was awarded to Hassabis and Jumper for this work. But that was only half the prize. The other half went to David Baker. And this is where our story gets really interesting. The protein designer. Remember David Baker, the philosophy major who wouldn't take "nobody knows" for an answer? While AlphaFold was about predicting protein structures, figuring out what nature had already created, Baker was working on something even more ambitious. He wanted to create proteins that nature had never imagined. In 2003, Baker's lab published a paper that should have been impossible. They had designed a completely new protein from scratch. They called it Top 7. Here's how they did it. Instead of starting with a sequence and trying to predict the structure, they ran Rosetta backwards. They started with a desired structure, a shape they wanted, and asked Rosetta to find an amino acid sequence that would fold into that shape. Think about how insane this is. It's like saying, I want to build a machine that can do X, and instead of looking at existing machines and modifying them, you figure out exactly what parts you need, in what order, to create something that's never existed before. Top 7 was 93 amino acids long. It had a structure that had never been observed in nature. And when they synthesized it and checked its actual structure using X-ray crystallography, it matched their design almost perfectly. Baker described it like this. If you want to build an airplane, you don't start by modifying a bird. Instead, you understand the first principles of aerodynamics and build flying machines from those principles. Top 7 was the right flyer of protein design. It proved that we weren't just limited to the proteins that evolution had stumbled upon. We could create entirely new ones. Since 2003, Baker's lab has created thousands of new proteins. Proteins that protect animals against the flu. Proteins that can sense small molecules. Proteins that self-assemble into molecular cages for drug delivery. Proteins that catalyze chemical reactions that no natural enzyme can do. One of his designs became the world's first computationally designed protein medicine, a COVID-19 vaccine developed at UW-Medicine. But even with all these successes, protein design was still hard. It required enormous expertise. It was iterative and time-consuming. Success rates were improving, but designing complex proteins with specific functions was still more art than science. And then AI changed everything. Again. RF diffusion, dreaming new proteins. In 2023, Baker's lab released something that sounds like science fiction: RF diffusion. RF diffusion uses the same underlying technology as image generation AIs, like DALL-E and Midjourney, the ones that create art from text prompts. But instead of generating images of cats wearing hats, RF diffusion generates proteins. Here's how it works. The AI is trained on thousands of known protein structures. It learns the patterns, the rules, the grammar of protein folding. Then, when you give it a specification, I want a protein that binds to this specific molecule, or I want an enzyme that catalyzes this specific reaction, it generates a completely new protein structure that should do exactly that. The diffusion part refers to how the AI works. It starts with random noise, essentially static, and gradually refines it into a coherent protein structure. It's like watching a sculptor start with a block of marble and chip away everything that doesn't look like the final statue, except the sculptor is an AI, and it's doing it in reverse, building up from nothing. RF diffusion can generate viable protein designs in minutes. What used to take months of expert work now happens while you get coffee. And in December 2024, Baker's lab released RF Diffusion 3, their most powerful version yet. It's 10 times faster than RF Diffusion 2. It can design proteins that interact with DNA, with small molecules, with other proteins, with atomic-level precision. It can create enzymes that catalyze chemical reactions with near-natural efficiency. The training code and model weights are open source. Anyone can download them, anyone can use them, anyone can build on them. As Baker said, when researchers from Sao Paulo to Nairobi can download the same code we use, that multiplies the rate of discovery. Improvements made by one lab benefit the entire community. The impossible becomes possible. So what can we actually do with these AI-designed proteins? Let me give you just a taste of what's already happening. Plastic-eating enzymes. Scientists have designed enzymes that can break down PET plastic, the stuff water bottles are made of, into their original chemical building blocks. These enzymes work at industrial scales, potentially enabling true circular recycling. Instead of plastic sitting in landfills for 500 years, it could be broken down and reused in days. Carbon capture. Researchers have designed proteins that can capture carbon dioxide from the atmosphere more efficiently than any natural process. We're talking about potentially pulling CO2 out of the air at scales that could meaningfully impact climate change. Next-generation medicines. Baker's lab has designed proteins that can bind to the spike protein of SARS-CoV-2, blocking it from entering cells. They've designed proteins that can target cancer cells with unprecedented precision. They've designed proteins that can cross the blood-brain barrier, something that has stumped drug developers for decades. Synthetic biology. Scientists have designed proteins that self-assemble into molecular cages, creating tiny containers that can deliver drugs to specific cells. They've designed protein-based nanomaterials with properties no natural material possesses. Biosensors. Researchers have created proteins that change color in the presence of specific molecules, creating cheap, portable diagnostics for diseases, environmental toxins, even explosives. And here's the truly mind-bending part. We've only scratched the surface. Evolution has had 3.8 billion years to explore protein space. It's found some amazing solutions, but it's also been constrained by history. Every new protein had to evolve from something that already existed. It had to provide immediate benefit to survive. It couldn't plan ahead. We can. We can design proteins with specific goals in mind. We can explore regions of protein space that evolution never visited. We can create proteins that are by any natural standard impossible. The rabbit hole goes deeper. Now, I promised you we'd go deep down the rabbit hole, so let's talk about what's really happening here, beneath the surface. The number of possible proteins is incomprehensibly vast. Remember that 10 to the 300 number from Levinthal's Paradox? That's just the number of possible folding configurations for a single protein sequence. The number of possible protein sequences is even larger. For a modest-sized protein of 100 amino acids, there are 20 to 100 possible sequences. That's about 10 to the 130. To put that in perspective, if every atom in the observable universe was a computer, and each computer could test 1 billion sequences per second since the Big Bang, we have tested basically 0% of the possible sequences. Nature has explored a tiny, tiny fraction of this space. All the proteins in all the organisms that have ever lived are just a drop in an ocean of possibilities. And here's where it gets really interesting. We don't actually know why nature chose the proteins it did. Evolution doesn't find the best solution. It finds a solution that's good enough, a solution that provides a survival advantage, a solution that can be reached through small modifications from something that already works. There could be proteins out there, proteins that have never existed in any living organism, that could cure cancer, solve climate change, create materials we can't even imagine.
And we now have the tools to find them. But, and this is important, we don't fully understand how these AIs are doing what they're doing. AlphaFold 2 doesn't think about protein folding the way a human scientist would. It doesn't reason about physical forces or chemical interactions. It's learned patterns from data, and it applies those patterns to make predictions. This is what makes some scientists uncomfortable. We have a tool that works spectacularly well, but we don't fully understand why it works. It's like having a magic wand that cures diseases, but we can't explain the magic. There's an ongoing debate in the scientific community about whether AlphaFold 2 has actually solved the protein folding problem in the traditional sense. It can predict structures, yes, but does it understand the underlying physics? Does it reveal the folding code that Ann Fenson hypothesized about? The answer, honestly, is not really. AlphaFold 2 is a phenomenally powerful tool, but it's not a theory of protein folding. It doesn't tell us how proteins fold, it just tells us what the final structure will be. And for many scientists, that's actually fine. As one researcher put it, we wanted the structures, we got the structures, let's use them. But others argue that without understanding the underlying principles, we're limited. We can predict natural proteins, but can we truly understand them? Can we predict how they'll behave in unusual conditions? Can we design entirely new classes of proteins that don't resemble anything in nature? This is where David Baker's work becomes crucial, because while AlphaFold 2 predicts what is, Baker's tools create what could be. So where is this all going? Let me paint you a picture of the future that's not science fiction. It's science happening right now. Personalized medicine. Imagine going to the doctor and getting a prescription for a protein designed specifically for your genetics, your disease, your body. Not a mass-produced drug that works for 60% of people. A precision therapeutic designed by AI, synthesized just for you, that targets your specific cancer cells or your specific viral infection. Programmatic materials. Imagine materials that self-heal, that adapt to their environment, that can be programmed to change shape or properties on command. These aren't science fiction, they're proteins. Protein-based materials that could replace plastics, create new types of electronics, build structures that grow themselves. Synthetic life. Imagine organisms, bacteria, yeast, even plants that have been redesigned at the protein level to do things no natural organism can do. Bacteria that eat pollution and excrete fuel. Plants that grow in deserts and produce medicine. organisms that can survive on Mars. Molecular machines. Imagine nanorobots built from proteins that can navigate your bloodstream, repair damaged tissue at the cellular level, clear out arterial plaque, destroy cancer cells one by one. These are being designed right now. But I also need to mention the flip side, because any technology this powerful comes with risks. What happens when anyone can design a protein? What if someone designs a protein that's harmful? A new toxin, a new pathogen, something we've never seen before and have no defenses against. The same tools that can design life-saving medicines can, in theory, design bioweapons. The same AI that can create plastic-eating enzymes could create enzymes that eat other things. This isn't to spread fear. The scientific community is very aware of these risks. There are biosafety protocols, security reviews, ethical guidelines, but the technology is advancing faster than our ability to regulate it. This is the double-edged sword of powerful technology. It can heal or harm. It can create or destroy. The difference lies in how we choose to use it. The story continues. I started this video by asking what happens when AI dreams up life forms that never existed. Now you know the answer. It cures diseases. It cleans up pollution. It creates materials we never imagined. It opens doors to possibilities that were quite literally impossible just a few years ago. But I also want to leave you with a sense of wonder about something else. For 50 years, the smartest people on Earth couldn't solve the protein folding problem. They threw supercomputers at it. They developed complex mathematical theories. They spent careers on single proteins. And then a team of AI researchers, people who weren't even biologists, who approached the problem from a completely different angle, cracked it in a way that no one expected. This is the power of bringing fresh perspectives to old problems. This is what happens when we combine human curiosity with machine intelligence. This is what becomes possible when we share our discoveries openly, when we build tools that anyone can use, when we democratize the ability to do science. David Baker has published over 640 papers, been awarded over 100 patents, and co-founded 21 biotechnology companies. 90 of his trainees have gone on to independent faculty positions. His dedication to open science has created a global community of researchers building on each other's work. The AlphaFold database has been accessed by over 3 million researchers in 190 countries. It's been used in over 7,000 published papers. It's accelerated science on a scale that was previously unimaginable. We're living through a revolution in our understanding of life itself, not by replacing human scientists, but by giving them tools that amplify their capabilities 1,000-fold. Not by solving biology, but by opening up new questions we never knew to ask. The protein folding problem isn't solved in the sense that we understand everything. It's solved in the sense that we now have a tool that works, a tool we can build on, a tool that opens up new frontiers of discovery, and the story is just beginning. That tangled mess of amino acids you see on your screen, it's not just a molecule, it's a story. 3.8 billion years of evolution, 50 years of human struggle, and now AI-designed proteins that never existed in nature, ready to change the world. The code of life isn't just something we read anymore, it's something we write, and we're just getting started. If you enjoyed this deep dive, hit that subscribe button. There's a whole universe of impossible problems out there, and we're just getting started solving them. Until next time, keep asking questions that nobody knows the answer to.